Welcome to section 8.2, Laws of Inheritance. In this section, we'll connect what alleles are to what we actually observe, practice Punnett squares, and make sense of Mendel's two core laws, segregation and independent assortment, and how they relate to meiosis. In this section, we'll build on Mendel's experiments to see how traits are inherited in predictable ways. We'll explore how genotype produces phenotype, how Punnett squares can be used to predict outcomes, and how Mendel's laws, segregation and independent assortment, explain what happens during meiosis. We'll also learn how test crosses can reveal hidden genetic information. By the end, you should be able to connect what Mendel discovered in pea plants to how inheritance works in all living things. A genotype is the set of alleles an organism carries. For example, it can be homozygous dominant, big Y, heterozygous, big Y, or homozygous recessive, little y. What is Y? Well, in this case, Y is the color of a pea. Um, big Y stands for yellow. Little y produces the green allele, but you only see the green allele if you have two little y's. So a phenotype is the physical trait we actually observe, like seed color. Genotype is the genes we inherit. Phenotype is the physical trait we actually observe. This distinction is important because two organisms can have the same phenotype but different genotypes. For instance, a yellow seed could be homozygous dominant, big Y, or it could be heterozygous, big Y. They're both yellow, but a green seed must be homozygous recessive, little y. So genotype is the genetic recipe, while phenotype is the dish that comes out of the oven. Mendel noticed that some traits mask the presence of others. We call these dominant and recessive relationships. In pea plants, yellow seeds are dominant to green. That means a single copy of the yellow allele is enough for the plant to have yellow seeds. The green trait only appears if both alleles are green. This dominant recessive pattern is simple, but not all inheritance works this way, which is something we'll explore later. Punnett squares are a tool for predicting genetic outcomes. By placing the possible gametes from each parent along the top and side, we can fill in the grid to see all possible offspring genotypes. For example, if we cross a true breeding yellow seed plant, where both alleles are dominant, big Y, with a true breeding green seed plant, where both alleles are recessive, little y, then all the offspring are heterozygous, big Y. The phenotype is all yellow, but the genotype reveals they are heterozygous. Punnett squares are powerful because they connect meiosis, fertilization, and probability into one diagram. We learned about Gregor Mendel in the previous section, and it's important to remember that he counted thousands of pea plants and saw these exact ratios appear, which is why his work was so convincing. Even without microscopes to see chromosomes, Mendel's math revealed the hidden rules of inheritance. Mendel's law of segregation explains why the 3-to-1 ratio happens. The law says that allele pairs separate during gamete formation, so each gamete only carries one allele for each gene. Mechanistically, this separation occurs in meiosis I, when homologous chromosomes pull apart. The important idea is that an offspring's genotype is the result of random combination, one allele from each parent. This explains why recessive traits can hide in one generation and reappear in the next. Sometimes you see a dominant phenotype, but you can't tell if the organism is homozygous dominant or heterozygous. That's where a test cross comes in. By crossing the individual with a homozygous recessive partner, the offspring reveal the hidden genotype. If all the offspring show the dominant trait, the unknown was homozygous. If there's a one-to-one mix of dominant and recessive in the offspring, then the unknown was heterozygous. This was a clever tool for Mendel and is still used today in plant and animal breeding. Mendel's law of independent assortment says that genes for different traits are usually inherited independently. This happens because in metaphase 1 of meiosis, homologous chromosome pairs line up randomly. That's what creates new combinations of traits in gametes. But there's an exception. If two genes are very close together on the same chromosome, they tend to be inherited as a package. This is called gene linkage. The closer the genes are, the less likely they are to be separated by crossing over. A human example is red hair and freckles. These traits are influenced by genes located close together, which is why they often show up together. This shows that while independent assortment is a powerful principle, the chromosome structure can modify how it plays out in real life. A dihybrid cross looks at how two different traits are inherited at the same time. For example, Mendel studied seed color and seed shape together. When he crossed plants that were heterozygous for both traits, he found a 9 to 3 to 3 to 1 ratio in the offspring. That means that 9 were yellow and round, 3 were yellow and wrinkled, 3 were green and round, and 1 was green and wrinkled. This ratio is the classic fingerprint of the law of independent assortment at work, because the alleles for color and the alleles for shape behaved independently during meiosis. It's like rolling two dice. The outcome for one die doesn't affect the other, but together they create many combinations. What's powerful here is that the dihybrid cross moves beyond single traits to show how complex combinations can arise naturally. It also highlights why sexual reproduction produces so much genetic diversity. Mendel's laws help us understand inheritance in a clear and predictable way. Genotype determines phenotype, but Punnett squares allow us to see the probabilities of each outcome. The law of segregation shows how alleles split during meiosis 1, explaining those 3 to 1 ratios. The law of independent assortment shows how genes on different chromosomes, or far apart on the same one, assort independently, unless they are linked. Test crosses provide a window into hidden genotypes. Together, these principles connect Mendel's pea plants to the molecular processes of meiosis.